I propose 3H hydrogen radioactive thymidine labeling of the plasmids during the triple transfection process for production of AAV, RAAV. This will make all the thymidine nucleotides radioactive. Recall, for lung cancer, in the triple transfection of our RAAV production, we use a Rep2/Cap5 plasmid supplied in trans. This plasmid has genetic elements of the AAV2 genome, chromosome 19 site-specific integration, and AAV5 capsid, lung specificity. The gene delivery system to be used will be adeno-associated virus helper-free system. For liver cancer, in the triple transfection for RAAV production, we use a Rep2/Cap3 plasmid supplied in trans. This plasmid has genetic elements of the AAV2 genome, chromosome 19 site-specific integration, and AAV3 capsid, liver specificity. The gene delivery system to be used will be adeno-associated virus helper-free system. Then the gene of interest with 0.864 kilobase DNA of E1A adenovirus is inserted in radioactive recombinant adeno-associated virus plasmid vector. Subsequent to triple transfection and RAAV variant production, the RAAV variant particles are delivered by injection into bloodstream, intravenous or intraarterial or intramuscular of cancer patient. The virus will integrate onto human chromosome 19 at a specific site, now causing portion of chromosomal DNA to be radioactively labeled. Upon expression of E1A adenoviral therapeutic gene of interest, apoptosis will be triggered in the presence of radioactive thymidine. As mentioned above, apoptosis will be high in cancer cells, lung and liver, while healthy cells will be better able to repair DNA damage caused by radiation, radioactive thymidine. Thus, the cancer cells in the lung and liver will be selectively destroyed using small doses of radiation at the nuclear DNA chromosomal level. Conclusion: we have developed a virus that can induce apoptosis at the nuclear DNA chromosomal level by selectively killing cancer cells, lung cancer and liver cancer, using a combination of radioactive thymidine and expressing E1A adenoviral protein, and this is useful for targeted cancer treatment. Insulin-resistant type 2 diabetes. Insulin-resistant is a physiological condition where the natural hormone insulin becomes less effective at lowering blood sugars. The resulting increase in blood glucose may raise levels outside the normal range and cause adverse health effects depending on dietary conditions. Certain cell types, such as fat and muscle cells, require insulin to absorb glucose. When these cells fail to respond adequately to circulating insulin, blood glucose levels rise. The liver helps regulate glucose levels by reducing its secretion of glucose in the presence of insulin. This normal reduction in the liver's glucose production may not occur in people with insulin resistance. In mice, it is found that endogenous glucose production is inhibited by purified recombinant ACRP30 protein. The mode of delivery is intraperitoneal injection. The study concludes that an acute increase in circulating ACRP30 levels lowers hepatic glucose production without affecting peripheral glucose uptake. Thus, a moderate rise in circulating levels of the adipose-derived protein ACRP30 inhibits both the expression of hepatic gluconeogenic enzymes and the rate of endogenous glucose production. In humans, the ACRP30 gene is located on chromosome 3q27. It is a 30 kilodalton protein. Thus, we can conclude that increasing the cellular concentration of ACRP30 protein shows promise for lowering endogenous glucose production without lowering peripheral glucose uptake, and this results in decreased blood glucose concentration, preventing the onset of type 2 diabetes. So, increased cellular